Hi everyone, my name is Carrie Lopez and I am going to be talking about antibody reagents today. So go ahead and open the Word document and y'all can follow along, take your own notes, just listen, whatever helps you learn the best. So, antibody reagents. Okay, now our antibody reagents are generally going to be, they red ones or clear ones? They're always going to be clear. However, sometimes reagents, sometimes they are dyed, like clear but dyed a certain color. So an example of this would be our anti-A. Is always blue. And our anti-B is always yellow. There we go. Now another important note I want to make is the difference in monoclonal or polyclonal and monospecific and polyspecific. So the difference in mono or polyclonal versus mono and polyspecific. So clonal is going to be in reference to how the anti-serum is made. And now your specific is what antibody here is directed towards. So for example, monospecific monospecific antibodies are specific to one antigen or epitope. Now examples of these are anti-IgG IgG or immunoglobulin G, or anti-C3d. So your key factor is going to be or. It's either anti-IgG or anti-C3d. Now your polyspecific antibodies can engage two or more different antigens or epitopes. Now examples of that are going to be anti-IgG or immunoglobulin G and anti-C3d. So they can engage both IgG and complement. So now we're going to talk about polyclonal. They are several different clones of B cells. So several different clones of B cells. And do y'all remember what type of cell a B cell is? That's going to be your B lymphocytes. Now the B cells secrete antibodies of different specificities. The mix of IgG, so immunoglobulin G, and IgM, so immunoglobulin N, has a mix of both of them in the reagent. How it's made: rabbits are injected with human red blood cells. Now, that being said, if they're being injected with human red blood cells, what requirement are you going to have to be able to manufacture this reagent? You are going to have a human donation. So you need a human donation to manufacture that. And so you think about that when choosing reagents and such, that's something that could play a huge role in their ability to manufacture efficiently. Now an example is going to be your AHD reagent or anti-human globulin reagent. And now these may be directed at different epitopes of the same antigen. So it recognizes multiple epitopes. And again, it may be directed at different epitopes of the same antigen. So now I'm going to insert this drawing I made for it, and hopefully it'll make it a little more clear. So here you've got your red cell. And this, I just made it a group A red blood cell. It doesn't really matter which kind it is. And over here, you've got one epitope. So that's a special epitope. And then this is a special epitope. And then this is another special epitope, for example, for one group A red cell. So these are the epitopes for surface antigen. Now, if you have a monoclonal, then it's just going to go against one epitope. So, for instance, just this epitope or just this epitope will be your monoclonal, but your polyclonal are going to target multiple epitopes. And so that can be, you know, just two or a mix of between all of them. So I did, for example, you've got this epitope and this epitope, or you can target all three epitopes, and that's going to be your polyclonal. Now, monoclonal antibody reagents. Monoclonal reagents are going to be your preferred reagent of choice. I'll make it even along with the other one that I start with. Okay, so it is made from single clones of B cells. So single clones of B cells. So B cells, antibodies, are they going to be the same or different specificity? They're going to be same specificity. Now, I'm going to make a special point here. Now, your monoclonal reagents are your preferred reagent of choice. This is because they are more accurate. It is more cost effective. Those are very valuable things in the lab. So accurate, cost effective, and if you remember before, what was required to make your polyclonal reagents, you needed a human donor. So this does not, your monoclonal reagents do not require a donor to manufacture. They don't need a human donor. Now, it's going to be, we talked about polyclonal being an IgM and IgG classes. So your monoclonal are going to be one immunoglobulin class, means it's either going to be IgG or IgM. They are manufactured in vitro, is that in the lab or in the body? That is in your lab. And then in vivo is going to be in your body. So these are manufactured in the lab in vitro using hybridoma technology. Let me go up here. I am going to put a little side note. I know we talked about specificity. Oh, we didn't. I did not include that. So I need to go up here and include it on the blood bank reagents video, but it didn't make it onto the antibody reagents video. So put down here. Are different kinds of standards that need to be met for licensure. And so one of these is potency standard, which is going to be your strength. So the strength of the reaction. And your other standard is going to be specificity. So it helps when we're talking about specificity to know what that's referring to. So that is the recognition of antigenic determinant and the corresponding antibody. So when we're talking about specificity over here, it's going to be the recognition of the antigenic determinant and the corresponding antibody. Now, what is a hybridoma? And hetero. So our hybridoma is the fusion of myeloma or tumor, kind of tumor, cells and antibody producing cells from one I want this all on the same page, so I'm going to put it down a little bit. So you've got two different choices. There's going to be murine. And now these consist of immunized mice. Then you mix the spleen cells with cells from a mouse with myeloma. Now I've got a picture that lays this one out a little better. Let's see. So you take your immunized mice, you isolate the immune cells, the antibody forming cells, you take those and mix them with a tumor cell, your myeloma cell, which fuses to make a hybridoma. We've also got human. Now, human hybridomas are formed when B lymphocytes transform by EBV. Now, does anyone remember what EBV stands for? That is your Epstein Barr. Now, some specific notes to go with your hybridomas. Oops. Now, since there are different kinds, referring to like murine versus human hybridomas, that means I always prefer the little arrow, like an equal sign with the arrow on it, to show like something happening, and so, or what something equivalates to. So, different kinds. Mean that you will have different strengths. And now that means, what can you do to know whether the strength of the reaction you are getting is the strength that you should be getting? The best way to find that out is to check the package insert. So, they will tell you whether the strength and the strength of the reactions or the potency of your reactions are what they intended it to be. Now, there are also heterohybridomas. These are human lymphocytes mixed with myeloma cells from a mouse. So mouse myeloma cells. An example of this is your anti-A, anti-little c, and actually, I want to clarify that this is part of your antibody reagents. So your monoclonal reagents. So let me go up here. I'll put them under here so you don't get confused. So an example is your anti-A, anti-B, little c. About some other ones. anti-IgG. And then I want to point out that anti-P3D is also monoclonal. And don't get it confused because, there's multiple letters there, but that's referring to complement as a whole, as a monoclonal reagent. They recognize single epitope so they have a unique specificity for one epitope. Unique specificity for one epitope. Now, one more time, what's specificity? Specificity is a recognition of antigenic determinant and the corresponding antibody. And then our little chart here, so for mono, it would just be one epitope. So it would react with one of those. And that's it for our antibody reagents. And next I will do one on all of your reagents together. Thank you.